Thanks for checking. You didn't miss anything. Don't worry. I was just introducing speaker, and what I was saying was, he's been an amazing mentor. He's actually a very sort of pressureful mentor to have, because every year he would write to me. And I would feel like, oh my God, this man is so busy and so productive and does all this amazing stuff and has the time to remember me. And so now I feel, so he's that kind of wonderful person. So in addition to a great scholar, I'm not going to tell you about his degrees and his qualifications and stuff. Obviously, he's very good. Otherwise, he wouldn't be invited to speak here. He's also an absolutely wonderful person. And when I found out he was moving to Providence, I was like super excited, feel like reunited after 30 years now. So it's good to have you here. And thank you so much.
Thank you, Shriram. Thanks for helping us get settled in Providence. So one of my best achievements is to co-author Shriram's first paper, in which he implemented and designed the checkpointing module for a software called FastLink, which enabled it to do runs that were far in excess of the expected time to failure of the workstations of the day. And that code actually, at least the design of that code, still survives. Does anybody here use the SciBlast module of BLAST? If so, I did the initial implementation, and one of the features of that module is that you can save the position-specific scoring matrix, and how you do that is based on Shriram's design. The code is now re-implemented by somebody else in C++, but the idea survives. And you can do the problem in reverse with a module called RPS BLAST that lets you search a set of position-specific scoring matrices. So let's plunge in. Oops. Okay. Let's see if that. Okay, what do I need to do to advance it?
Maybe, okay, I bet I know what happened. When I asked Zoom, I gave permission to Zoom, it maybe focus shifted away from Zoom. Let's try that and now try again. Oh, this isn't even on top. Is there an AV person here?
There we go. Okay, great. All right, so let's plunge in. The talk is in two parts. One is about the past of how clinical trials in oncology have been conducted and which trials have a particular structure. And the second part is about the future and some speculations about how oncology treatment will be delivered in the future. The unifying theme of the two parts is that we ask questions that start with how many. For those who study algorithms or statistics or epidemiology, those questions are quite familiar and comfortable. My experience working with biologists is that biologists are more focused on mechanism. I won't say too much about mechanism today, but when studying mechanism, the questions usually start with what and where and why. But to do things like the NIH Cancer Moonshot, you see, we're going to have to be able to answer questions that start with how many. Okay, so for the first part, we're going to ask how many oncology clinical trials have particular structures. We've done 4 such projects so far, and I'm going to talk about the last 3 on this slide briefly each. One is how many utilize germline information. Second is how many use the principle of synthetic lethality, which I'll explain when we get to it. And the last is how many make comparisons of outcome or side effects between males and females. Most importantly, here are the project participants. My supervisor, Dr. Eitan Rupin, he gave a talk at your cancer center on June 1st, 2023. I promise there's 0 overlap between what the projects I'm speaking about today and the 4 projects he spoke about on that date. Student trainee who was first a senior and then a postbac, Ashwin Kamula, a former Rupin Lab member, Ju Sang Lee, who's now a faculty member in Seoul, Korea. Sheila Rajagopal, oncologist and computational scientist in the Rupin Lab, and Roselle Kurzrock, one of the world experts in oncology clinical trial design. So I like to say a little bit about how projects started to give the students in the audience some ideas. So in this case, Eitan and Ju had the idea to study clinical trials of immunotherapy, and I'm doing that now. And to do that, Eitan's budget had paid for a single user license of a resource called Trialtrove, which I'll explain in subsequent slides. But then Ju left for a position in Republic of Korea, and the license had already been paid. So I had taught formal languages in the 1990s based on this textbook, which may be familiar to some of you. And so I thought, okay, this is a chance to use my formal language training, and you have to pay for the license, so there's not going to be too many competitors. So this is a good project for me. Luckily, nobody else in the lab volunteered, so I got to be the single user. And the project formally started on May 5, 2021, when I got some syntax training in how to use Trialtrove. Okay, I like to start my talks with a quote from a research giant. And for this talk, I picked a quote from the late, great Janet Rowley, famous oncologist. Does anybody here collect stamps or have a child who collects stamps? No? Okay, I guess it's out of fashion. Well, she was a serious stamp collector for her whole life, and she said that probably I was good at it, meaning science, because I had spent years and years looking at stamps in very fine detail, watermarks of paper, and a Scott stamp catalog. And what she's famous for is identifying chromosomal translocations that characterize various blood cancers, including most famously chromosome 821 translocation that characterizes some forms of acute myelogenous leukemia, and chromosome 922 translocation that characterizes some forms of acute lymphocytic leukemia that brings together the genes BCR and ABL and constitutively activates the kinase ABL. And you can read about that achievement if you like popular science books in this book, The Philadelphia Chromosome. Her discovery was a stepping stone for Brian Drucker and colleagues to develop the drug imatinib, which was the first effective kinase inhibitor in oncology. So the first part of the talk highlights curation, and we'll talk about kinase inhibitors and related treatments in the second part, and that's the connection. So we're going to talk about Trialtrove. So what is it? It's a curated collection of over 400,000, essentially all human interventional clinical trials for all diseases, but we're going to focus on the oncology subset. It's a mixture of structured data and free text. There's a user interface, which I won't show. You can download the data, which is important to freeze the data and do large-scale data analysis. You can ask questions, and if you report errors, they will actually fix them because you're paying. Okay, I'm a US government employee, so I shouldn't be seen as endorsing any one product. So I'll say there are at least 3 competitors. And of some interest, the first competitor, Cortellis, is made by the same company that produces Web of Science. So if your library was generous, you may actually have a license to Cortellis in addition to a license to Web of Science. Our library doesn't. Okay, so you might have heard of another free resource from NIH called clinicaltrials.gov. Why not just use clinicaltrials.gov? So there's several reasons, including the structured data and the curation, but this study showed that Trialtrove, because it uses many sources from around the world, has many more trials than clinicaltrials.gov, in addition to more information about individual trials. Okay, so when I start a project, two questions to ask are the results going to be publishable, and who's the competition? And in this case, the publishability is important because you're using a licensed product and you can't do a data dump, so there might be some issues with some journals. So I had to Eitan assigned me to find papers that use Trialtrove and also to find the competitors. So there are some previous papers that use Trialtrove, and our key competitor is Professor Andrew Lowe at MIT, who works at the interesting interface between business, drug development, and AI. And you can see here's a highly cited paper of his that relied on Trialtrove. So we're good to go as long as he's not doing the same thing we are. All right, now let's get into the details and follow the guidance of my favorite character from Sesame Street, let us count. Here's one snapshot of how many trials of various sorts there are in Trialtrove. We freeze the data roughly every 6 months, and the listing how many trials treated patients of both sexes is important for the third part, where we're going to look for sex comparisons. You can see by comparing the numbers that the number of oncology clinical trials grows by roughly 5,000 per calendar year. Okay, so we're going to plunge into the first question. How many trials use germline information? One reason I love questions that start with how many is you get a number. That's the answer. Now you can argue about the methods, what to include, exclude. The number is 887. That is a small enough number that I could curate every single individual trial and make sure that there are no false positives. There may be false negatives, but no false positives. Okay, first question we can ask is, what are the tumor types? By far, the most common tumor types are breast cancer and ovarian cancer. I'll explain why in the synthetic lethality part. For what follows later, notice that colorectal is third, non-small cell lung cancer is fifth, and lymphoma is eighth. The acute lymphocytic leukemia that Professor Rowley worked on is 10th, and acute myelogenous leukemia that she worked on is 15th. Okay, this is the $64,000 or $64 million question. If you use germline information in the design of your clinical trial, are you more likely to get a positive outcome than if you don't? And the answer is yes. By far, you'll see in a later slide that the way we counted positive and negative outcomes, the rate of positive outcomes is in the 40s. And I know that seems high to some, but remember, many of these are phase 1 trials that are usually successful because people have done animal studies for safety. But importantly, among these germline trials, there are quite a few phase 2 and above trials. So this germline information is already having clinical impact, especially in breast and ovarian cancer. And so there should be more trials using germline information. I'll give some examples shortly. All right, so here, next question is, what about the genes? So consistent with breast and ovarian cancer being the top cancer types, the hereditary breast and ovarian cancer genes, BRCA1 and BRCA2, are at the top, along with some other breast cancer genes. Anybody want to guess why BRCA2 has more trials than BRCA1? Any clinicians here? Okay, so the answer is that germline variants in BRCA2 also are associated with susceptibility to pancreas cancer, but that's not true for BRCA1, so that explains why some trials look only at BRCA2. I want to talk about 4 surprise entries in this top 20 list that surprised me. First one on the positive side is ERBB2, that encodes the protein HER2. You may have heard about Herceptin, treatment for HER2 positive breast cancer. So I knew that ERBB2 is important at the protein level, but it turns out that there are germline variants that are also important to the response to Herceptin. Similarly, for EGFR, I thought that's important because there are somatic mutations in non-small cell lung cancer and other cancers. But again, it turns out that there are germline variants that affect its expression and response to EGFR inhibitors, which we'll talk about later. On the negative side, surprising to me was MSH2 and MLH1 at the bottom. These genes are mutated in a hereditary cancer syndrome, sometimes called Lynch syndrome, sometimes called hereditary non-polyposis colorectal cancer, which manifests usually as colon cancer or endometrial cancer in females. It's very important to look for these germline variants because it's known, and this has led to FDA approval, that immune checkpoint inhibitors are especially effective in this cancer. So there should be many colorectal cancer trials which pick out the 5% or so of patients that have Lynch syndrome and treat them differently. And there are some trials, but not nearly enough. So the supplement of our paper has many genes that should be pursued more. And finally, here's another sad story. There's major geographic health disparity in where these germline trials are conducted, mostly in the United States and Europe. And that's very unfortunate because it's missing many opportunities to leverage variants that are predominantly seen in other parts of the world and affect susceptibility to hereditary cancers. Let's move to the second part, which is about synthetic lethality. So any students here from Professor Larchan's lab? Okay, anybody here studying Drosophila or yeast? No? Okay, so synthetic lethality is well known in Drosophila and yeast. It's a relationship of two genes here depicted, A and B. And the idea, as shown in the left slide, is that the relationship between these genes is that if you knock out or inhibit one of the genes here shown as B, but it could be the same for A, the cell survives. But if you knock out or inhibit both genes A and B, then the cell dies. And this is very useful in cancer therapy, and this was first suggested by the Nobel Prize winner Leland Hartwell in a 1997 paper by the following paradigm. Suppose you have a cancer that has the gene B mutated, think BRCA1, and you can find a synthetic lethal partner for that gene. If you can inhibit the other gene, what will happen is that the cancer cells die and the non-cancer cells survive. And so we looked for trials that have this design, but we generalized a bit to other trials which use two drugs, one that inhibits A and another that inhibits B. Of course, to do this, we had to curate. Remember the importance of curation from Professor Rowley's quote. We had to curate hundreds of established synthetic lethal relationships. Okay, so again, here's the answer to the how many question. There were 1,207 oncology clinical trials using this definition of the principle of synthetic lethality, 754 using the more standard biomarker design, 618 in some sense using the two drug design, although 165 can be found either way, and that gives you a total of 1,207. I was very surprised that this was higher than the number of germline trials. Okay, again, we can ask about cancer types and genes. So here you can see in the cancer types, now lung cancer rose to the top, breast is second, lymphoma has risen to third, and so forth. In terms of genes, here we have to look at gene families, and this is why breast cancer is so near the top. The discovery that thrusts synthetic lethality into its current clinical importance is that if one inhibits at least some members of a family of human genes called PARP that are involved in repairing certain kinds of DNA damage, then that's a synthetic lethal partner of BRCA1 and BRCA2. So people with BRCA1 and or BRCA2 cancers are now frequently treated with PARP inhibitors. It's a bit complicated because the different PARP inhibitors inhibit different subsets of the genes, but we won't get into that. We'll just lump them together. Here, the two interesting genes I want to talk about a little bit are KRAS and TP53, well-known cancer genes. I had been to, before the discovery of KRAS inhibitors, I had been to dozens of talks where KRAS and TP53 were labeled as undruggable genes. And that's just false, because each one of these has a synthetic lethal partner, at least one. And there are many CDK4, CDK6 inhibitors FDA approved, and there's one WEE1 inhibitor approved. So you can find quite a few clinical trials that propose to give adavosertib, the WEE1 inhibitor, to patients who have TP53 mutations. Again, the $64 million question is, if you use the synthetic lethality principle, is your clinical trial going to have a higher probability of success? So the answer is yes, although it's predominantly because of the ones that use the traditional design, as you would expect. If you give two drugs, that doesn't really leverage synthetic lethality. So you could argue that part of what's going on is people are just repeating the PARP inhibitor trials over and over, and that's why they're successful. They're just fine-tuning the details. So that's true for PARP inhibitors. The difference between those trials and others is very significant, but even for all the other trials, not PARP inhibitors, it's still a significantly higher probability of success. We documented in our paper well over 100 synthetic lethal partners that are tested in mice and have not been tested in a single human clinical trial. So there's plenty of opportunities for clinical oncologists. Let's move on to the third part, sex differences. Here we're asking not about the design, but what's reported in the literature. And we were quite surprised and disheartened that we could find only 472 clinical trials that reported where Trialtrove had a curated comparison between males and females. There's a strong publication bias, so about 2/3 represent differences. The similarities are omitted, and we documented why. Those usually get buried in the supplemental information, and Trialtrove typically doesn't curate that. Okay, so importantly, a large majority of the trials that have differences are favorable for females, and as you'll see in subsequent slides, that's all about the survival, response, and outcome. For side effects, there are more trials that are favorable in males. Okay, so in our paper, we characterized 4 levels of evidence: multivariate analysis, univariate analysis, general numerical comparison, and just qualitative statement. So if you focus on the ones that actually have statistical analysis, the difference is even more striking: 122 to 47 trials having a better outcome in females. Okay, so some people have argued that this is just because in general females have better survival in cancer, but that's not true. I mean, it is true that females have better survival in cancer, but that doesn't explain what we're seeing. What we're seeing is largely because of 2 diseases: non-small cell lung cancer and lymphoma. And even more interestingly, it's because of a disease-drug interaction. Come back to EGFR here. The difference in lung cancer is mostly explained by trials in which the drug was an EGFR inhibitor. For other trials, there's not much difference between males and females. And here, these 2, if you look by treatments, these are the first 2 generations of EGFR inhibitors. The third one explains the lymphoma difference. Rituximab is an antibody that inhibits CD20, which marks B cells, and rituximab is a very effective treatment for non-Hodgkin's lymphoma, and that entirely explains the difference, the favorable outcome for females in lymphoma, but the mechanisms are not known. The females responded better to EGFR inhibitors was known. There's been some speculation about the mechanism. No answers. For rituximab and lymphoma, there was one anecdotal report, but no systematic analysis before our paper. And again, it's a disease-drug interaction because rituximab is given often for autoimmune diseases. And although there are some anecdotal reports in autoimmune diseases, there's not a clear advantage in outcome for females receiving rituximab. So for those of you studying mechanism and biology, here's 2 opportunities to study important questions at the boundary of signaling and oncology. Okay, now we dig into those details that Rowley told us to look at. Why are so few sex comparisons published? So to set up this question, I'll point out that publishing comparisons by sex is required by US law, amended in the 1990s. So this predates by more than 20 years the NIH's widely publicized policy on sex as a biological variable, which is relevant for non-clinical trials, but the law is not being followed. So what's going on? All right, so now we need to look carefully. I'm going to pick on the Lancet family of journals today, but the same applies to New England Journal of Medicine and Journal of Clinical Oncology. Here's one trial published in Lancet Oncology, and you do see analysis by sex in what's called a forest plot, and you might look at hazard ratios and see, okay, for females, 1.0 is outside the confidence interval, so this is a significant improvement. For males, 1.0 is inside the confidence interval, so this is not significant for males. That's all correct. And then you might take the next step and conclude that the response, in this case, progression-free survival, is better in females than males. Anybody want to tell me why that inference would be wrong? Okay, it's wrong because no direct comparison was done. Each subgroup was analyzed separately. Here's another paper in a Lancet journal. Again, same situation. 1.0 is in the male confidence interval, not in the female confidence interval. But in this case, if you look at the fine print, a comparison was done and it's not significant. Okay, look at the fonts. Don't they look similar? What's going on here? Lancet makes so much money from the publication charges that they can have their own in-house software that redraws the forest plots. And so every forest plot of subgroup analysis in every Lancet paper looks exactly the same in terms of font and layout, but they don't enforce that the comparison is done for subgroups that are comparable. All right, so in summary, let's start here. If the journals required comparisons, which is easily implementable in software, right, Shriram? We could easily increase obedience to the law and the number of male-female comparisons. Both germline trials and synthetic lethality trials have a higher positive outcome rate than other trials, but they excessively look at BRCA1 and BRCA2 and breast and ovarian cancer. And there are many, many opportunities to look at other genes for germline trials and other pairs for synthetic lethality trials that are yet to be pursued. Okay, let's move to the second part, speculate. Yes. So those differences in those trials are basically random. The sample sizes are small. Basically the sample size. I mean, so one of the things we did indirectly in our paper was to find how many trials have sample sizes of over 200, and it's very rare. We could find fewer than 100 clinical trials that had sample sizes of over 200. So it's small sample size. Usually not, because so that's another problem, right? Cancer incidence in males is much higher, especially for lung cancer, and so they typically enroll by incidence, not to power for a comparison of males and females. So in lung cancer trials, you'll get easily 5-to-1 male-female ratio. Yes, we should, but it's all I'm saying is it's just that's random. Yes, yes, we should. Yeah, absolutely. No, it's an interesting question in terms of can you reverse engineer those forest plots to judge which comparisons are likely to be significant. So that's a pure algorithms question or statistics question. So, oh yes, the question was basically, why do you see those differences in those trials? Can we explain it? Are they real? Are they random? And my answer is they're basically random, having to do with small sample sizes. You can see the ends here are in the 50s and 60s. So in this trial, there were 68 males and 55 females. Okay, let's move to the second part, which is about speculations on future therapies for cancer. So the motivation is that we believe in the future, and it's already happening in the present, the effective therapies for cancer are going to be combination therapies, because that overcomes intra-tumor heterogeneity, genomic differences between different cells in the tumor. And so the key data we're going to use in this study is single cell transcriptomics data to model the effects of future treatments on single cell transcriptomics data, we're going to have to define the space of possible treatments and define what it means for a treatment to kill a cell. I will do that in subsequent slides. So here's the explicit research questions. I'm going to focus on the second one. A patient walks in and has single cell transcriptomics done. Can you estimate for some class of drugs how many drugs will be needed for a typical patient? And in particular, we'd like to identify the two ends, the patients for which 1 or 2 treatments will be effective, and the patients for whom you need 10 or more and it's hopeless, you shouldn't even try. And then for the algorithmicists here in the audience, I'm going to focus on an interesting multi-layer optimization problem. How can we balance the objectives of treating each patient in a personalized, optimal way versus treating a cohort that we need to run a clinical trial to show that our ideas might be effective? Okay, so I should say there's related prior work from Brown, and I'm glad Eliopho is here. He worked with Fabio Vandin and Ben Raphael on these 2 papers and others using optimization problem called set cover to study problems in oncology. We're going to study a related problem called minimum hitting set to study some other problems in oncology. And this is a chance to thank Ellie, who in 1986 recruited me for a student internship at IBM Almaden, which was a very formative experience in my career. Okay, so we're going to talk about specific numbers of treatments in later slides. So I want to set some baseline. Some patients treated for lymphoma receive a regimen that has a single name called R-CHOP, but if you look under the hood, they actually receive 6 drugs. Some patients with solid tumors, and this is by far not the extreme, receive a regimen called FOLFOX. Again, they give it one name to make you think you're getting 1 drug, but you're actually getting 3 drugs. And I also want to pay tribute to my employer. Combination therapy was pioneered at NIH by these 5 individuals in the 1950s who laid the basis for modern chemotherapy. And for that, they shared the 1972 Lasker Prize. Okay, again, the most important slide, the project's participants. The work is guided by my mentor, Eitan Rupen. The implementation was done by Saba Ahmadi and Pat Sukrasert, who were computer science students thinking they were doing theory under the guidance of the theoretician Samir Kuller, some who recognizes Samir's name here. So he's now the chair of computer science at Northwestern University. 3 former members of the Ripon lab, Rahul Vagesna, Sanju Sinha, and Theo Shishlik, helped with data analysis, curation, and visualization. And we started, remember to record the start date, October 30th, 2018, based on a question posed by Natalie Artsy at Brigham and Women's Hospital. And it's somewhat relevant for what follows later, that Natalie studies, she's a chemical engineer, and she studies nanomedicines. And I'll explain why that's relevant in a later slide. Okay, for the computer scientists here, this was my perspective on the problem. We know for decades that good software is modular. Can we use a modular approach to treat cancer? And what would that mean? What it would mean is that we have to solve 2 problems. We have to recognize the cancer cells and we have to kill them. Let's suppose, at least for this theoretical analysis, that we're going to reuse the same killing mechanism over and over, but what's going to differ from one drug to another is the recognition mechanism. Another concept from computer science that we used is we want to kill the cancer cells, we want to spare the non-cancer cells. So this is a trade-off, like time-space trade-offs we study in theory. Okay, and we formalize this trade-off as follows. We set a baseline lower bound on the proportion of tumor cells that must be killed, and an upper bound on the proportion of tumor cells that can be killed. You might think that this 0.8 is low. Some clinicians we've shown this to think it's low, but actually, if anybody's familiar with something called RECIST criteria, which are commonly used in oncology, they're interested, for starters, in a partial response, and you can make a geometric argument that a partial response corresponds to roughly a value of 0.7. So we're actually being stringent, and in one subsequent slide, we'll be more stringent. Okay, so I wouldn't be focusing on the modularity if it wasn't already in practice. Here's one commonly used treatment called CAR-T, which is particularly effective for B-cell cancers. And you can see it's modular. modern CAR Ts have 5 modules. I'm going to focus on the 2 extreme ones. On the outside is an antibody that recognizes a cell surface receptor on the surface of the cancer cell. And on the innermost part is starting with CD3 zeta, a trigger to the signaling cascade that triggers CD8 T cells to kill cancers. Important to the development of CAR T has been these middle modules, but I'm not going to talk about that. Okay, CAR T is not alone. There's at least 4 classes of drugs, conjugated antibodies, and CAR T are already in clinical use. The other 2 are more hypothetical, that have this property that they have a recognition subunit and a killing subunit. Now, there are fundamental differences. Key to what follows is that in conjugated antibodies and nanoparticles are based on the concept of overexpression in the recognition subunits. They recognize proteins that are overexpressed, whereas CAR T recognizes proteins that are expressed at all. And we'll model the overexpression shortly. So here's the workflow for our software. Cohort of patients comes in, has single cell RNA transcriptomics done. We record it in the conventional way where rows are genes, columns are cells, except we split the cancer cells from the non-cancer cells into files. And then here's our software. The secret sauce is integer linear programming, which is familiar to some of you, to solve variants of a classical NP-complete problem called minimum hitting set, which I won't explain. And out comes the optimal treatment for each patient, or an optimal treatment for each patient. And the idea of the way we draw the pills here is that the white part is the killing subunit, which is homogeneous for all the pills. And the shaded part is the recognition subunit, which is different from one pill to another. Okay, how do we model that? I'm going to explain that in this slide. Thank you. Okay, so I said that nanoparticles and conjugated antibodies are based on the idea of overexpression. So the recognition subunit works by binding to proteins that are overexpressed on the cell surface and are receptors, so that the drug can get into the cell via receptor-mediated endocytosis. The physical chemist Don Frankel and his disciples have worked out a theory called the theory of superselectivity, or now hyperselectivity, which they have tested in vitro and a little bit in vivo. And I have a bunch of slides in case anybody wants to learn about that, but I'm not going to talk about it otherwise, to show that you almost get a sigmoid function. Above some threshold, you will recognize the target, and below some threshold, you generally won't. Now, the problem is that ratio is gene specific, but we don't know it. It's only been measured for a few genes. But based on some data, we reasoned that a small single digit number of 2 is reasonable. We went from 1.5 to 5 in our different experiments. So what does this ratio 2 mean? Remember, we split the non-cancer cells and the cancer cells. So we take the average of the expression of the non-zeros for the non-cancer cells. We say that's the baseline average expression. Any cell that expresses more than twice that is going to be killed. So if we pick ALK as the target, you take 6 plus 23 plus 4, divide by 3, that's 11. So any cell that expresses more than 22 units of ALK is going to be killed. And so it's not a great target. We'd kill 1 tumor cell and 1 non-tumor cell in this example. KDR is a much better target. We'd kill 2 out of 3 tumor cells and not kill the non-tumor cells. Okay, and this is how the thresholds work. We're going to accept any solution that's within the thresholds, but we won't accept solution that violates either threshold. Now let's get to the answer. Yes? Each are single cells. Yes, exactly. And we ignore the zeros in the non-tumor cells because they may be dropouts. Okay, so now we can answer the question. How many treatments are going to be needed per patient? We're really saying how many targets are needed per patient. We studied 9 data sets. I ask you to trust me that these are the 4 most complex ones. And you can see the numbers, often 1, surprisingly, or 2. But for some patients, the x-axis is the patients, the y-axis is the number of targets needed. For some patients, it's close to 10. The variation here is because we thought we should do some sampling and show how many cells we need to get a stable answer, but the software can handle all the cells. Okay, so in case you didn't think that 0.8 and 0.1 are stringent enough, we did the same analysis with 0.9 and 0.05, and you can see for some patients the answer doesn't change, but for other patients it doubles. So the stringency matters for some patients, not for others. You really have to, the oncologist has to decide what should be an appropriate target level of cell killing. Okay, in this analysis, we justify our existence. We compare the optimal solution to a greedy solution. That's how combination therapies are currently designed. And you can see that the patients are lined up, so they're the same in the same order. And you can see, for example, for these patients on the right-hand side in the melanoma data set, the greedy solution is more than double the size of the optimal solution. So optimization can work in practice. Okay. Of course, we should find the optimal solution personalized for each patient out of respect for the patients. But in reality, we have to run a clinical trial, and that's going to have a cohort. So can we combine those two aspects? And this is for the algorithmicists in the audience. How can we do it? So two ideas. The first one on this slide. Suppose in this toy example, we have two patients, A and B, and they each have optimal solutions of size 2. Why should we pick the optimal solutions on the left? Anybody? Reuse. Specifically, we're saving the pharmacy money because the pharmacy needs to only order 3 drugs instead of 4 drugs. And Ellie's laughing, but that's real because in a real clinical trial, the menu of drugs is not all FDA-approved drugs. It's a limited menu. So we want to reduce the cost to the pharmacy. Okay, so we actually, in the software, solve a 2-layer optimization problem among all the solutions that are optimal for single patients. We find a solution that's optimal for the cohort as a second layer, and that hadn't been done for hitting set. It had been done a little bit for traveling salesman problem for the theoreticians here, and in the supplement of our paper, you can see related work in optimization. No, that is not it's still NP complete. It's not above NP complete. It's just a double minimization because you can encode it as separate terms. Yeah, good question. Okay, so let's talk numbers, not just theory. How much can we benefit from reusing targets? In the head and neck cancer, if you sum the optimal solutions, it's 13. By reusing targets, we get to either 4 or 5, depending on the replicate. In the melanoma data set, averaging over 20 replicates, if you sum the size of the solution, 54.4, reusing drugs, 31. That is a substantial savings for the pharmacy. Okay, now a second idea of balancing cohort versus patient, which fits into the area of algorithmic fairness. And I know there's a competing event today about fairness in AI, so I'm talking about fairness too, at least on this slide. So we decided to model fairness in this way. Suppose that we allow the possibility that some patients will not receive an optimal treatment, and we model that with an extra parameter alpha, which is an upper
not receive an optimal treatment
for that patient. They would receive more drugs. Yes. So they get more drugs in return for increasing the drug reuse. So they might get 3 drugs instead of 2 drugs, but that enables the pharmacy to order fewer distinct drugs.
Is there a clinical reason to not do that?
To do that or to not
do that? Because any harm,
you might kill them because the drugs interact. Okay, and they might be unhappy if they learn that they get a suboptimal treatment. At least if they're theoreticians. Okay, so those are 2 reasons. And maybe the treatment would be less effective. Okay. So there are reasons. Yes, there are consequences, but this is a trade-off, and this is theory, and we're just talking numbers, not actual treatment. So we can theorize. Okay, so this parameter alpha, the way you should read this is increasing alpha goes down the y-axis. And what you want to ask is, what's the smallest alpha at which you reach the optimum? So for the brain cancer data set, it's 1, and you can reduce the average number of total treatments for the cohort from 10.6 to 9.8. The numbers for melanoma are different just because we just did a different random seed. Here you can the optimal alpha is 4, and you can reduce the number of drugs the pharmacy has to order by 10. And in some sense, this is fair about fairness because it allows more patients to participate in the trial for the same pharmacy budget. Okay, for the biologists in the audience, they might be interested in the target sets of size 1. What are those special receptors? So in 3 out of the 4 brain cancer data sets, the phosphatase PTPRZ1 showed up. We're not the first ones to suggest that targeting PTPRZ1 is a good idea, but it hasn't made it into clinical practice. We couldn't persuade anybody to do the experiments, but fortunately, a research group in Switzerland read our paper, picked up on the idea, and made a CAR T for PTPRZ1. It's still in preprint form, posted last December. Hopefully, it'll be published. And I won't go into the details, but if you're familiar with Kaplan-Meier curves here, they showed the mice treated with PTPRZ1 CAR T do have better survival. So it looks like it's a reasonable idea. If you want to do some biology, here's an open problem for head and neck cancer. We found, and another computational group also found, that the orphan G protein coupled receptor, GPR87, is a good target. It's been shown to be overexpressed in various tumor types, but barely studied. What orphan means in this case is that it's a receptor, so it's on the cell surface, and nobody knows what the natural ligand is. So there's an open biology problem for any of you who study G protein coupled receptors. Okay, let me wrap up. Most patients in our modeling will receive single-digit treatment combinations, an average of less than 3 in our baseline setting, which we know based on the full Fox and R-CHOP examples is a reasonable number, but there's a long right tail, and we should not give false hope to those patients. An optimal combination can be much smaller than a greedy combination. And for the algorithmicists here, we define 2 generalizations of hitting set that might be interesting in other contexts, where you have overlapping instances with the same universe, and where you allow a certain amount of suboptimality and you study the trade-offs. The paper is published and our software is freely available. Let me conclude by saying the software is called Mad Hitter, which was Eitan's idea to combine hitting set with the Mad Hatter character from Alice in Wonderland. I like to conclude by saying the Mad Hatter helped Alice look through her looking glass both forwards and backwards. And in that spirit, we're looking backwards at how clinical trials have been done in the past to understand what worked better and what worked worse, to make some speculations about how oncology clinical trials should be done in the future to give personalized therapy. Thank you very much for coming, and I'll take any questions.
Thank you for that wonderful talk. Are there any questions in the audience?
I'm kind of thinking about your problems here and your minimization and trying to line things up. There's reminds me because we use Gurobi for this activity too, or something like Gurobi.
Yeah, so we use Gurobi.
I noticed where the problem we're trying to deal with is like you're doing configuration of like secure systems. And you have to like flip switches, and like in military systems, they're all physical switches, and you have like 700 of these, and you can't expect somebody to accurately do 700 configurations. So what you're trying to find is like, what is the minimal set of configurations that'll get you to what you want? And you in particular want to know what the don't care states are, because those you don't have to set at all, and you want to minimize the number you want to set. And it's the same sort of like double quantification, because you can do this, but then if you know the current state of the system, you want to minimize relative to what you've already got.
So yeah, I think the way to think about it in that context is, suppose you have the universe of all switches that exist in these systems. Can you define instances with overlapping sets of switches where you can somehow reuse setting the same switches in location A and location B, so that you get a reasonable solution, even if it's not optimal. So the closest and this is the practical example that has been studied in theoretical computer science, is for traveling salesman problem. Basically, what UPS and FedEx would like to do is have a sort of standard set of routes that on most days will deliver most packages pretty efficiently, even if they aren't optimal for that day. Exactly. It learns only a finite set of routes and chooses them on each day. So that's been studied. It's sometimes called universal traveling salesman problem. We could not use the term universal hitting set because that's been used in another computational biology paper for a different problem, so couldn't use that term, but that's the idea. And so we think this idea has potential outside of this problem, and we're already using it in another cancer project. So hopefully some computer scientists here will popularize the idea. Yes.
Explain how your treatment combinations sort of like the two ways combinations that be effective for the one like this is the one single patient where emphasize or the single drug or treatment of population where the single patient one of the drugs in the combination is most effective right now.
Right. So this is yes. So the question is, why do we think this way of approaching combinations is effective? Is it dealing with heterogeneity or is it dealing with resistance or both? So it's dealing with heterogeneity. There is a school of thought, I won't say it's dogma, that if you hit the cancer right away with the logical OR of a bunch of treatments, you will kill the different clones, the different cells before they have a chance to develop resistance. We're not dealing with resistance here because to deal with resistance, you would have to change the killing subunit. So that's you could reuse the same ideas, but we can't do that with the pre-treatment single cell transcriptomics data. You need some post-treatment data. And actually, one of the projects that Dr. Rupin talked about, the perception project that I'm also involved in, we did study resistance and looking on how to use single cell transcriptomics data to adapt the therapy over time as resistance emerges. So again, we think single cell transcriptomics data is the way to go, at least until there's widespread single cell protein data. But resistance is a different problem where you have to change the killing subunit, and you can keep the recognition subunit the same unless the cancer cell becomes resistant by shutting down the cell surface protein, which does happen. So great question. Yes. Yes. In traditional greedy search, you would add one treatment at a time. So you would decide that an EGFR inhibitor is effective, and then you would add an ALK inhibitor, and then you would add a different kinase inhibitor, and so forth. You would add one at a time. We actually implemented adding two at a time because some people do that, and even adding two at a time is not great. You really have to explore the exponential size space. So the difference is we are considering all combinations simultaneously, even though it's a huge number. There's been 50 to 60 years of research in combinatorial optimization that gives us powerful tools like Gurobi that can do that. Oh.
Exactly. So you can say the competitive is not. That means I might say the driver is not.
Right.
This is like
Right. So I want to take that in three levels because it's a great question in three parts. So the question is from Ellie expressed in a kind of computer science style, but really has clinical impact is that, we're assuming that if you add either in the greedy formulation or the optimal formulation, a fifth drug to a 4-drug combination, that can only benefit. But in fact, there might be side effects and so forth. Okay, so let me take it from what we modeled and then in general. So this is where the modularity is key. We're assuming that the killing subunit is the same. And it's only the recognition subunit that differs. So when you give 5 pills here, you're really giving 5 instances of the same toxic drug. You're not giving 5 different toxic drugs. And that's key. And we other real oncologists doing these sorts of trials recognize that. The problem with drug interactions is when you give different drugs that act on different pathways in their toxicity, and that can be very beneficial. That's called synergy, or it can be disastrous, and that's the antagonism that you're referring to, and other people in the Rupin lab study that. It turns out that a very small fraction of drug pairs are synergistic, so that those magic combinations are very hard to find. And that's where the somehow attacking 2 different pathways simultaneously is beneficial, not just because it blocks resistance, but there's actually some molecular interaction. We're not going for that. We're not so ambitious. Here, we're using the same killing subunit over and over. So that's why we think it's monotone and additive, but we agree that you might have to adjust the dose. There is a little bit of chemistry assumption here. When we're giving different targets, they have to find the different molecules on the cell surface, and the recognition subunits might collide. So some chemistry modeling has to be done. You're right.
It's not even monoclonal, it's item.
Yeah.
A second one.
Yes, but you may have to adjust the dosage of the killing subunit so that the drug, the patient doesn't get too much total drug. Yes.
I think I missed something where if you're using this in trial setting, if you use computer science modeling, aren't you going predictive modeling anything?
No, because it's patient specific. So we're not, this is not a machine learning problem. So there's a set of trials. They're mostly using mutation data. A few trials, Roselle, Kurzrock, whom I mentioned, led a trial called the Winter trial, which was the first one of this type to use RNA data. So these are so-called basket trials. What that means is the pharmacy has a basket of available drugs that is much larger than typical trials, tens of drugs. Patient comes in, some omics is done, and the patient receives a treatment based on the patient-specific omics. Now, there may be false positives in omics and so forth, but in general, they use, except for the very early trials like the Winter trial, they're using validated biomarkers. So the biomarker should work for that patient.
Okay, so you're trying to find competition for patient, not
Yes, exactly. The sense in which the cohort comes in is we have to have a trade-off to keep the budget down.
In the interest of time, let's give a round of applause to the speaker. And thank you everybody for joining us. Right, so you have meetings tomorrow.
I do.